Hello.
चलो हाँ ठीक है ना, ओके।
हाँ क्लास सर
यूट्यूब पे आ गया लाइव?
आ गया लाइव।
हाँ सर।
Sir, should we start right at 4 o'clock, sir?
Wait, I think wait for 4 or 5 minutes.
Okay.
हेलो हेलो, अरे हो हेलो हेलो हेलो।
आय कार्ड घेऊन जा ना, मग आय कार्ड घेऊन जा. पकडलं तर आय कार्ड दाखवायचं ना, कॉलेजला गेलो होतो ना. का बरं? I think Pochar.
Yes, sir.
Hello.
Yes, sir.
We can start, no problem. It's 4 o'clock around.
Right, sir. Right, sir. Correct. Devani, sir.
Yes, sir. Start करते हैं. Yes, good afternoon to all the participants. A very good afternoon to all the participants in India, and good morning to all the participants who are joined from US. I, Anil P. Devani, Assistant Professor P. Wadhwani College of Pharmacy, on behalf of management, principal, and staff of P. Wadhwani College of Pharmacy, Yavatmal, welcome you all to the second day of this international webinar that is organized by our institute, that is Pharmacy 360, Research to Practice. On the first day, that was a day yesterday we had our first day of this webinar. On the first day of the program, we had Dr. Chaugule as a speaker with us, who discussed about the aerosolized nanomedicine-based drug delivery in lung cancer. It was a very, very interesting talk that was delivered by Dr. Chaugule to us. Now for today's session, that is the second day of our webinar, international webinar, we are having with us Dr. Subrata Dev from Larkin University, College of Pharmacy, USA, to deliver his valuable talk on the topic Pharmacogenomics in drug disposition, case of COVID-19 investigational drugs. So it will be a very, very interesting talk that we will be hearing from Dr. Subrata Dev. We are also accompanied with Professor Madia sir, who is from Institute of Pharmaceutical Education Research, Wardha, who will be chairing this session as a chairperson of this session. Now, from here onwards, I hand the proceedings to Dr. D.S. Mohale for giving the brief introduction of Dr. Subrata Dev. Dr. Mohale, please.
Okay. Thank you, Devani sir. Am I audible?
Yes.
Okay. Good afternoon to all. I am Dr. Deepak Mohale, Associate Professor, P. Wadhwani College of Pharmacy, and coordinator of this session. On behalf of management, principal, and staff of P. Wadhwani College of Pharmacy, once again I welcome you for this international webinar series, Pharmacy 360 Degree Research to Practice. Here I am to introduce today's speaker, Dr. Subrata Dev sir. He has completed his M.Pharm in medicinal chemistry from Bombay College of Pharmacy, Mumbai. He has completed his PhD in pharmaceutical sciences from Faculty of Pharmaceutical Sciences, the University of British Columbia, Vancouver, BC. He has completed his postdoctorate from Vancouver Prostate Center at Vancouver General Hospital, Vancouver, Canada. He has completed certificate course in Foreign Pharmacy Graduate Examination Committee conducted by National Association of Boards of Pharmacy, USA. He has worked as teaching assistant at Bombay College of Pharmacy, Mumbai, a research associate, analytical method development at Indigo Remedies Limited, Mumbai, a research scientist, drug discovery and development at Torrent Pharmaceutical Limited, Ahmedabad, graduate teaching assistant, graduate research assistant, and vice president administration at University of British Columbia, Vancouver, Canada, clinical instructor, Faculty of Dentistry, the University of British Columbia, Vancouver, Canada, assistant professor, Department of Pharmacy, Tripura University, Tripura, India, problem-based learning instructor, Faculty of Medicine, the University of British Columbia, Vancouver, Canada, a research associate, Vancouver Prostate Center, Vancouver General Hospital, Vancouver, Canada, assistant professor, Biopharmaceutical Sciences, College of Pharmacy, Roosevelt University, Schaumburg, USA, and presently he is chair and associate professor of Department of Pharmaceutical Sciences at College of Pharmacy, Larkin University, Miami. He has published more than 27 research and review articles in peer-reviewed journals and newsletters. A book chapter having title Drug Metabolism, Detoxification, and Xenobiotic Biotransformation is accepted for publication by Apple Academic Press. He has presented more than 54 presentations in various international conferences, of which 19 were invited presentations. He chaired more than 19 conferences. He has attended many international conferences, workshops, and seminars. Uh, he is recipient of fundings from many universities and professional bodies. Some of them are Prevent Cancer Foundation research grant of $1 lakh, National Institute of Health T35 training grant $4,73,000, Roosevelt University intramural grant $12,500, and many more. He is also recipient of many awards, including Michael Smith Foundation Postdoctoral Fellowship Award, Jack Carlisle Founder Award, Douglas and Jean Bailey Scholarship, Early Investigator Award, Just Desert Award, and many more. Apart from this, he is also running orphanage at Tripura, India. He is serving as a reviewer and editorial board member for many reputed journals. This is, in short, biodata of Dr. Deb sir. Now I request him to proceed with his talk. Thank you.
Before proceeding, myself, Dr. Chandevar, Principal, Pivadwani College of Pharmacy and convener of this international webinar. I would like to invite Dr. Subrata Deb for his presentation, and I am very much thankful for his kind acceptance. I talked to Dr. Prashant Sakharkar and requested him to talk to you for giving the presentation, and immediately you accepted that one. For which I, on behalf of my management, Yavatmal Jilla Vikas Samiti, I am very much thankful to you, sir. I am very much thankful to chairperson of the today's webinar, Dr. D.K. Madia, sir, being a chairperson. Thank you, Madia, sir. You have also accepted our request and for being the chairman of this station. Thank you. Thank you very much. Over to Mohale.
Okay. Dave sir.
Yes, good morning. Am I visible and audible here?
Yes, sir, you are visible as well as audible.
Okay.
Good morning.
Great. Good morning. Good afternoon, everyone. And thank you, Dr. Anil Chandevar, sir, for contacting Dr. Sakharkar, and then he approached me. We worked together for some time, Dr. Sakharkar and I. So I thought it will be a great opportunity to connect with your college and community. And I would like to thank, before I start, to Dr. Chandevar, Dr. Kocher, Dr. Shirao, and Dr. Mohale for very kind introduction. I'll start my presentation, and I'll be always happy to answer any question or discuss anything at the end of the presentation. Okay, so I'll be sharing my screen now, and we'll go from there. Okay, so the slides are now visible, right, from my end?
Yes,
Yes, sir.
Okay, so if I can request everyone, please to put the microphone on mute so that there is no feedback for any of the participants. That's the microphone, if you can please put on mute. Thank you. So before I start the topic, because typically when Dr. Sakharkar and I come to India, we try to visit some pharmacy school in summer, especially. That's the time we have opportunity, and I thought this is a good time because of COVID and everything. I think the world has changed a lot in between, and this is, I think, a very good effort by P. Wadhwani College of Pharmacy in bringing people together and still focusing on task at hand by the pharmacists and everyone involved in healthcare. But before I start my topic, I just wanted to briefly give some background of where I belong to, and some of them you already heard. And thanks to the technology, we are sitting about 9,000 miles apart right now. And we still feel connected. And I think I can't imagine in early 90s or something, or something like that, or even when I left India about 20 years back. So technology has brought us together, and we still feel connected even we are far apart. And a bit of my roots in India and where I lived before moving, I belong to actually Tripura in Northeast. I did my schooling from Agartala, Umakanta Academy. That was my schooling. That's in Northeast part, extreme Northeast part of India. Then I moved to Odisha, where I did my undergraduate in Baharampur. That's a town closer to Andhra Pradesh, near Vizag. And then subsequently, I was in Bombay College of Pharmacy in Mumbai. And I'm hoping there are some alumni in the audience, at least one or two, if not many, from BCP, Bombay College of Pharmacy. A very fond memories of those days in Bombay. And eventually I moved to University of British Columbia, Vancouver. But I think my connection to Mumbai is still there, and I still visit Mumbai every time I visit India, which is either each year or every other year. So with that in mind, I would like to start the conversation here. The goal of this presentation or talk is really to provoke some research thoughts. I thought at this time there are several unanswered questions in our mind, and some of them are definitely related to COVID-19. And the whole world is trying to grab whatever solution they get, be it vaccine, be it existing medication repurposing. But of course, we have mixed reaction, mixed outcome of those drugs when people used over last 4 months. And some of the failures, some of the success could be explained by pharmaceutical science technologies and concepts. I see a hand, Dr. Kirti Bhushan Barik. I think there is a question or something. Hello.
No, sir. Question answer session will be at the end of your talk.
Okay.
So-
So I'll request the participant accordingly. If you could please unraise the hand on Zoom. So thank you. So essentially, the goal of this conversation is we start thinking, okay, what kind of thing we can do as a pharmacy community or pharmacist or pharmaceutical technologists to bring a better solution, not only just COVID-19, and also down the line similar diseases, because the world disease spectrum has changed. 50 years back, bacterial diseases were more of a bigger problem, right? But now the problem is shifting more towards viral diseases. Of course, oncology is always a problem. Dr. Jogale talked yesterday about lung cancer. So cancer has been always a problem, but viral diseases are getting more common now compared to 50 years back when bacterial diseases were the problem. So moving forward, the objectives of this talk will be first a very brief understanding of cytochrome P450 enzymes, drug metabolism, and its relationships with pharmacokinetics. How these superfamily of enzymes contribute to PK parameters, and then I'll be introducing the importance of pharmacogenomics in translational drug metabolism, means how we can take it from the lab to the patient and vice versa. And then subsequently, potential role of pharmacogenomics in COVID-19 treatment. And finally, what research opportunities are available right now in translational drug metabolism. This is my sincere hope that the first three objectives will eventually generate or raise interest among the audiences here to pursue some COVID-19 related or translational metabolism research either now or down the line. So very briefly, I'm sure we have seen as a pharmacy student at some point or a pharmacy teacher at some point, this is a typical flow of drug action. Like, for example, once we administer the drug, it gets absorbed, distributed, and eliminated. These three processes are vital before the pharmacological effect can be exerted. But our interest today is in this component, drug metabolizing enzymes. So how drug metabolizing enzymes contribute to drug action, because you realize that drug concentration at site is very much dependent on what is going on in the ADME area, the PK area. So ADME essentially controls how much drug will be reaching to the site of action and eventually causing either clinical response or toxicity. Either of them are possible, irrespective of we want or not. So our focus, drug metabolizing enzymes. And in that, the purpose of drug metabolism, we realize, is making drug molecules from lipophilic to hydrophilic components. And once they're hydrophilic, they are easily excreted in the body, from the body through urine and so on. So the fundamental reason we break down, we biotransform, or we metabolize, we can use any terminology. The bottom line is some chemical changes happening in the body biochemically, through which we produce water-soluble molecules that leads to excretion through phase 1 and phase 2 enzymes. And in terms of liver, any metabolism which occurs in the body eventually reduces drug bioavailability. And if you think about how drugs are circulated, once we take the oral medication, it goes through portal vein in the liver and then it goes to the liver. Here in the metabolism pathway occurs first time, and then through hepatic vein it goes to systemic circulation, and then again it comes back through hepatic artery to the liver. So you can see liver getting exposure twice, and that's the terminology first pass effect comes from. And the outcome of first pass eventually is reduction of drug bioavailability. That means the lower drug concentration in plasma. The opposite is also true. If the drug is not getting metabolized in the liver, the bioavailability is going to increase. Okay, and this is a typical PK parameters curve where it is affected by drug metabolism. And if our goal always is, be it any disease, keep the drug in the therapeutic range between the MAC and MTC. The goal is to bring the drug within this window so that there is no toxicity, as well as there is no lack of therapeutic action or therapeutic failure. And you can see typical PK parameters such as area under the curve, Cmax, all these are or could be affected by drug metabolism because they deal with concentration. Anything which deals with concentration is going to be affected by metabolism. Now, CYP enzymes, like I said, it's one of the major class or superfamily of enzymes which is present in liver and small intestine, the two major metabolic sites. And they are present in endoplasmic reticulum as shown in this, these blue colored bags. And their goal, and they metabolize every chemical you see on this earth, drugs, environmental toxicants like pesticides, cigarette smoke, you name it. Endogenous substances such as vitamin D, that is one of my research area actually, which I'm working for last 10, I think more than the 12 years. endogenous substances like vitamin D, hormones, they are also metabolized by CYP enzymes. So this is a vital class of PK modulator. Now, you may have noticed in different textbooks, different papers, they name like, very differently, unlike other enzymes. They say CYP2D6 3A4, right? But look at other enzymes. We say esterases, angiotensin converting enzyme. But cytochrome P450 or CYP, they are named very differently because, and I'm going to say it here, when you say human 2D, CYP2D6, human is the species, obviously. CYP is the superfamily. And then the next numerical number is actually family. They have several family and subfamily. Just think of our society, how we are divided in different families and so on. And then finally, you have another letter, which is actually subfamily under the family 2. And then eventually the numerical number 6 tells individual protein. So you can think about number 6 as an individual, right? We all are different, but eventually we are part of some microfamily, and that those microfamilies are part of bigger extended family, right? And so on. And then eventually we are part of on humankind. So likewise, it's a very complicated to some extent, but that's how we name all nomenclature these CYP enzymes. And these are done based on amino acid sequences. They are classified in family or subfamily. So, and among the phase 1 CYP enzymes, the 4 is always very critical because they metabolize about 90% of the drugs in the clinic. CYP2D6, which we will discuss a lot today, CYP3A4, and 2C8, 9, and 2C19. These 4 are the major CYP enzymes which metabolize about 90% of the drugs we see. Now, one of the challenges with CYP enzymes is they can be either induced or inhibited. Means they can be their expression level, protein expression level can either be increased or lowered or blocked. Now, this is an example what can happen when you give St. John's wort. It's a herbal product very much available in United States and other countries, Europe, for seasonal depression. But that drug is not prescribed, by prescription, so that is easily available in the retail stores. Now, when you give St. John's wort along with verapamil, then they increase the metabolism of R-verapamil or S-verapamil, the stereoisomers. So if you look at the first curve on the top, the open square one, this is without any St. John's wort, just the drug itself. But as soon as you added St. John's wort, you will see about 5-fold difference in the concentration of verapamil. So the CYP3A4 levels are increased. Here the levels are increased by St. John's wort. As a result, the concentration of R-verapamil is decreased. So remember at the beginning I said drug metabolism has an effect on PK levels, Cmax level, AUC levels. So this is what is happening by increasing CYP3A4. The metabolism of R-verapamil is increased. As a result, the verapamil blood concentration is decreasing. And that's why you see that drop of concentration when you combine them. Same thing is also applicable for S isomer. You can see at the bottom of this curve. But really, the impact on R-verapamil is very strong, 5-fold difference between single versus combination administration. On the other side, when you have the grapefruit juice given with felodipine. So felodipine, again, as an antihypertensive metabolized by 3A4 enzyme. And grapefruit juice is a strong 3A4 inhibitor. And the first line, if you look, second line in green is without the juice. But as soon as you give grapefruit juice, the concentration again increasing more than 5-fold, and you can see here in the left hand side in the y-axis. So this tells that felodipine metabolism is blocked by grapefruit juice, and eventually the concentrations are increasing, which can lead to hypotensive side effect and even patient discomfort and so on. So these were two examples of induction or increase or decrease of metabolism. Now the next phase where I'm going to make the transition is about translational component of drug metabolism research, because in the lab everything is good, we can do all kinds of stuff, but eventually we want to translate into human treatment outcome. And if you look at this simplistic diagram, in the left hand side you have influencing factors, and there are several listed here, like drugs, herbal medications, disease, diet, everything. Then the drug metabolism in the host, means in humans, and then all this metabolism happening, and eventually drug response are being affected. And even microbes, you can see there is a magnifying glass in the middle diagram. The microbes also contribute how our metabolism is controlled. So in other words, microbes can control how our cytochrome P450 levels are expressed. Most cases they downregulate, means most cases they suppress the expression of CYPs. So it's not only different drugs and diet, also biological factors such as diseases, such as COVID, for example, can actually lower the CYP expression, means lower drug metabolism, and eventually you may see higher blood concentration of the drug, means potential for toxicity. Now, I'll be changing the gear a bit and more towards now pharmacogenomic aspects. I'm sure we have always seen that between our friends, relatives, colleagues, we have seen this type of question. Okay, this drug is working for you, but not for me. It works for my mother, but not for my mother-in-law. So these are very common questions, be it a pharmacy individual like us, or be it a layman person. We all ask these questions. And there are several reasons, obviously, why there is a difference between drug response in individuals. But one of the most prominent one and scientifically proven for last 10-15 years, this is a new area, of course, is genetics. Otherwise, we always say age, maybe your body type is this, you may have some other disease. So we always give these answers, which are true, which are definitely contributing how we respond to a drug. But at the same time, genetic composition of individuals is one of the major contributing factor. And interestingly, a long time back, Hippocrates, who was a physician, philosopher a long time back, he said, I think it is important to know the person before we prescribe anything, more than knowing the disease. If some of you have got any experience with homeopathy from the past, they always ask questions. What do you do? What your family does? What type of habits you have? Symptoms you have? So they rely on individuals' experience or try to understand the person more than the disease or diagnostic tests, which is very opposite to allopathic approach. But the bottom line is to get the optimum outcome of a drug treatment, we need to realize an area called pharmacogenomics, which is a combination of two terminologies, pharmacology and genomics. And if you combine them, it becomes pharmacogenomics, essentially the study of genes in drug response. And this diagram gives an idea how the whole population is different. how we all need different kind of dosing for optimal response, how we can predict toxicity ahead of time, not after people experienced it. For example, in this diagram, if you look at in the left hand side, this is a common population, and you can see they're color coded. Some are gray, some orange, some purple, and in the right hand side, you can see they are segregated at the bottom also. So this profile is good. They are going to get favorable response. This is the most of the population which we expect. The purple color one, that's a genetic profile for non-response, means the individuals are not going to get outcome from this drug. There will be a therapeutic failure in these people. And if you look at the orange color at the left and bottom side, their genetic profile for toxicity means they are going to get severe adverse effects that will lead to discontinuation of the drug or potential death. So you can imagine, we can all understand this even before the drug is administered if we use the technology or science behind it. The goal is eventually to identify people who are going to get favorable response, so that you only give the drug to those conventional individuals, not the other one. For either financial reason, it will be a economic loss, as well as a health loss for genetic profile, for toxicity of the individuals. So take home message is, one size doesn't fit all. As we all have different needs, this science drives the treatment to individualized or personalized medicine. Now, of course, my focus will be cytochrome P450 area, but now along with 2D6, can you see something like star allele or star 5? So the first component remains the same, like I said earlier: superfamily, family, and subfamily, and then protein. However, at the end, for CYP alleles or variants, you use the word star 5. You use the word star 5, and the star nomenclature tells you that it's not the same as wild type, means it's not the same as common people. If some individual has star 5 in their genotype, that means that person will have a different metabolic profile. So because this person has nucleotide variations, means the amino acid sequence of that person has changed. And this slide is going to tell you what does that mean. Okay. So if you look at this diagram, this is the gene from the same person, and it's called single nucleotide polymorphism, or SNPs. Means one nucleotide is changing, just one, and that leads to a major change in the protein component. And this is the wild type if you look at the top structure. Then if you look at the person 2 in the second structure, there is one nucleotide change from alanine to guanine. But the amino acids didn't change, because in our body there is redundancy, means the same protein can come from 2 different amino acid sequence. But look at the last one, person 3. Because of change from guanine to adenine, the amino acid has changed from arginine to lysine, and also the structure of the protein has now changed. So as a result, you anticipate different protein expression and different activity. And in terms of identifying different pharmacogenetic variants, how do we do that? For example, you have a patient population of 100 people. You take their saliva or blood and do genotyping through different molecular biology techniques, including RT-PCR. Then eventually you can find genetic types which will be people for toxicity or non-response, or people who will have normal response to drug therapy. So you can segregate people based on their genetic composition. and this is eventually the outcome you want to see, that okay, I have a population where the amino acids changed or the nucleotides changed, and some of those are responders in green coded I put here, and some of the non-responders. And eventually you can find an association between the genotype and the drug response phenotype, means what you are observing. And that will lead to identification of population which are either responsive or non-responsive to the drug treatment. And in the process, you have identified a genomic marker, means a marker which can suggest the success or failure of the drug. And this is essentially summarizing what I have said so far, that drug metabolism polymorphism occurs, means change in sequence that lead to amino acid sequences of the C protein, and then that means modified or no enzymatic function that leads to change in PK parameter change, and leading to altered efficacy and toxicity, which will require either dose adjustment, means you need to lower the dose to avoid toxicity, or totally remove the drug from the patient. So this is kind of a sequence of events usually we want to see from the lab to the patient, and modify the treatment plan according to that. Now, again, this one just gives you another idea, another glimpse that that alleles can change drug concentration. So x-axis you have time, y-axis you have concentration, and on the top you have different genotype. You have either wild type homozygous, wild type heterozygous, means two combination of two things, not the same allele, because each gene has two alleles in our body. So they can be either same or different. So first one wild type, second one wild type heterozygous, and third one polymorphic or modified form variant homozygous. Now, if you look at the AUC of this, there is a 2 to 4-fold difference as you go from left to right, because you have now modified enzyme. You don't have the same enzyme as the genotype at the beginning, wild type. And as a result, you see increase in AUC or PK. That means that is going to lead to that means that is going to lead to more toxicity or more patient death, especially in the last category, where you are looking at 4-fold difference of PK numbers. And eventually, these enzymes or these genotypes are categorized in 4 phenotypes, means something you can observe. For example, hair color is a phenotype. Skin color is a phenotype. Means because of genomic changes, we are seeing some effects on the we are seeing some outcome in the metabolism. So they are classified as poor metabolizer, means they cannot metabolize the drug at all. Intermediate means they're average. Extensive, their metabolic rate is high. And then finally, ultra rapid metabolizer, means they metabolize drugs very quickly. So these are the class of patients we categorize: poor or PM, intermediate or IM, extensive or EM, and UM. So I'll be using this terminology subsequently to highlight that either these people can't metabolize or they metabolize very rapidly or something in between. That's called the phenotypic representation. And if you think about it, if you think about it, some of the concept here, think about a funnel, a glass funnel in the lab or something. I am not sure if you see some arrows. I'm not sure where from they are coming. Do you see some arrows in between some of the different color arrows?
I can see error.
There was an arrow.
color.
There was an arrow. I'm not sure if how it is coming on the slide, because I'm not drawing them. So I just wanted to make sure. So So poor metabolizer is a poor metabolizer is a class of people who can't biotransform the drug, can't filter the drug. As you can see, all these medications are stuck on the top of the funnel, so they can't come out and get excreted. On the opposite, normal metabolizer, they come through the funnel and they're filtered. Eventually, they are excreted in the urine. So that's the difference. You can think in your mind. This poor metabolizer are a funnel where the drugs are stuck there; they cannot come out. And in case of normal metabolizers, they essentially metabolized and eliminated and filtered out of the process. And this is the difference between the phenotypes. And again, to bring back the original diagram, we had poor metabolizers, they have decreased metabolism. That means they increase the concentration above MTC. They increase the concentration to toxic level. That's why I coded in red, because if you have decreased metabolism, that is going to lead to more death or toxicity. Whereas in case of increased metabolism by or ultra rapid metabolizers, the concentration is going to come down to the bottom in the MSC, below minimum effective concentration. As a result, there will be therapeutic failure, means patient is not going to get the therapeutic outcome. So you see, both the extremes are bad, be it poor or be it ultra metabolism. Both of them are going to impact either by increasing more death and toxicity, or by lowering the concentration to less toxicity, I mean less therapeutic outcome. So so the next the next one is about CYP2D6. CYP2D6 is one of the major enzymes, as I mentioned, that it is responsible for metabolizing very important class of drugs, antidepressants, antipsychotic, analgesics such as codeine, and most importantly, a lot of cardiovascular drugs, antihypertensive drugs are metabolized by 2D6. And there are more than 100 polymorphic forms, means in the population, if we measure today, there will be 100 different types of enzyme isoforms. That means it eventually going to impact the metabolic, and the frequency of those alleles are different between different population. And if you look at this table, which highlights that there is phenotype frequency, means how often you see them in Asian, African American, and Caucasian. These are the three major ethnicity in the world. So if you see the frequency, or means the availability of different CYP phenotype, you will see Caucasians are highest when it comes to either poor metabolizer or ultra rapid metabolizer. Whereas Asians are also affected by CYP2D6 polymorphic forms or variant forms. So this is an example to show that unless we do some studies, we don't know what is exactly the population frequency in our area. For example, in Indian context, the phenotype or the genotype of people could be very different in western part of India versus northeast part of India, because of the origin of individuals. And I'm going to give some more thought about this towards the end, but this is just to highlight, this is not just internationally phenotypic differences. Even within the country, there are differences of genotype and phenotypic frequencies between different population. And this is an example how dose can be different. So nortriptyline, we all know, is an old drug for as an antidepressant. Now you look at the bottom, the dose, the range of dose is needed dose is 50 milligram to 500. there is a tenfold difference of dosing needed. Now, for most of the people, you need about 150 to 100. But there are people where you need 20 to 50 only, or where you need more than 250 or 500. So you can see the range is so wide. If we give the people who need 50 them 500, it will be a catastrophic effect, and vice versa. So it shows that the genotype is related to phenotype, and then eventually it is related to dosing. That means how much drug you want to give. And if we don't give appropriate dose, of course, that's the reason we always get some unexplained deaths. And sometimes we say, nobody knows what happened, right? This is a very common phrase in medical. But of course, there must have been something in the physiologically, pharmacologically, something happened which led to the death, perhaps from toxicity of the drug. And in terms of practice, currently clinical guidelines in this area, and you can visit at your own leisure, this one. This is the website on the top of the slide, CPIC, cpicpgx.org, Clinical Pharmacogenetics Implementation Consortium. So there's the forum in the United States supported by NIH and other agencies who develop guidelines how to use those pharmacogenomic information into clinical guideline, pharmaceutical guideline. And all these guidelines are actually freely available on PubMed. If you go to PubMed and type CPIC guideline, and in the left-hand side, as you can see here, I have made that red box. You write practice guideline. If you choose that filter, then you are going to see all these free articles and freely available articles, and you can apply in clinical practice, drug development, and all different kinds of research in the lab and so on. So the last part of it, which I'll be introducing before I conclude, is the current situation, the COVID-19. And I'm not going to give much info about the disease number and everything, because I think we are watching them day in and day out on TV, news channels, and newspapers. So as of yesterday, we have crossed 12 million worldwide. So this is probably the biggest, I'll say, challenge to humankind after Second World War to the modern history. Now, if you look at this complex curve, which shows almost all countries which have COVID-19, top 3-4 countries, here US is on the top. This is an unfortunate situation, but we are at the top of this. But India is also there on the top 3. Actually, it's not coming out because you can see that there are 2 curves here. Russia and India are very close by. So India name is not appearing on the curve, but India is right now, I believe, is 3rd in number. So both US and India are actually literally struggling right now how to deal with it. And I believe pharmacists have a big role in voicing their opinion when it comes to drugs and investigational drugs, because there is no approved drugs for that particular disease right now. So as such, the coronavirus or COVID-19, this particular viral strain, it enters through the use of angiotensin converting enzyme 2 receptor in pulmonary area, and then eventually it goes to whole body, including liver. So there might be a thought that it only remains to lung. That's not the case. It goes to liver, it goes to kidney, it goes to, now even we know it is also attacking brain. So that means it's also impacting drug metabolism because it's infecting liver. Think about hepatitis. We all know about that, right? So hepatitis affects the liver, and we know hepatitic patients' liver metabolism is very different than a normal person. But when it comes to COVID-19 patient, all the focus is right now on lung. But a major area people are not thinking, literally not thinking, and I check every day for any new paper, new grant, or new information. Uh, there is absolutely no thought process right now that this is also affecting your liver. And if it is affecting your liver ability to metabolize drugs, there are other implications we are going to say in a minute. And some of those we started talking in the earlier half. So, but just to give a quick view, what is available and what people are juggling with right now as a solution of drugs in the clinic. None of them are obviously approved for COVID. Uh, there is all kinds of drugs in different component, either targeting the virus before entering, or they are targeting the translation or viral replication process. All this, out of these, two drugs I think worldwide causes some huge interest. One is hydroxychloroquine, right? I'm pretty sure we all know that. And second one is remdesivir, which is from Gilead, which has got some very good data actually, by the way, a few weeks back or last week actually. Remdesivir and hydroxychloroquine, these are the two drugs perhaps found the most acceptability or interest in the clinic. And hydroxychloroquine will be my focus for rest of the talk. Hydroxychloroquine actually blocks a fusion step. When the virus enters the body in the host cell in the lung, they need to get fused to because you know that virus doesn't have its own replication system, right? They can't replicate by themselves. They use the host system to replicate. So the COVID-19 virus, when it enters, they need to get fused. Membrane fusion need to occur with the human body. And that's the step hydroxychloroquine is potentially blocking. So you watch my word, potentially, because it is not proven, that nobody has done experimentally. These is all people have hypothesized, and all the treatments are right now going on based on hypothesis. And that's the reality we are facing today. Now, hydroxychloroquine, it's an old drug, and I'm sure all of us, when we were in the BFARM days, this was the one of the very much studied drugs for malaria, right? Uh, Although it was discovered look at the timeline, how old this drug is. But you will be surprised and maybe sometimes sad that in spite of that, it is an old drug. There hasn't been a lot of research you expect from such a good anti-malarial drug. About 10 years back, people started using it for for things like some inflammatory diseases, lupus, or autoimmune diseases. But again, research is limited in terms of non-anti-malarial diseases. And now we are in a situation we have to use a drug without much information in humans, because anti-malarial use, there was not much choice that time. So people did use even with lot of clinical evidence or pharmacokinetic evidence. Now, both chloroquine and hydroxychloroquine is a 4-aminoquinoline derivative. And that's why you can see the only difference is in their side chain. I don't practice medicinal chemistry anymore, but I did my MPharm in medicinal chemistry from Bombay College of Pharmacy with Dr. C.L. Viswanathan sir. So I still have some interest in chemistry, how molecules look like, although I don't talk a lot. So hydroxychloroquine and chloroquine are very similar, but the hydroxy derivative found more interest in the clinic and more effect also, if anything, in the clinic. Now, this part is now tying back to what we talked initially. Hydroxychloroquine is metabolized by CYP2D6 and other enzymes, as you can see here. You follow this path in the right-hand side. Hydroxychloroquine is metabolized to different subsequent steps by CYP2D6. And remember, I mentioned CYP2D6 has at least 100 or more type of variants in the population. So it has a different implication, means the way we metabolize hydroxychloroquine is very different. Even if I think about the participant here who are in this conversation, we all have different ability to metabolize hydroxychloroquine. And another thing, another issue with this particular drug, it has a very high volume of distribution, means it remains in the body for a long period. And if you look at the half-lives, it is like 45 days, plus minus 15 days. So I remember in my BPharm days, and I'm sure all of you have studied as well or looked at some point, Brahmankar's biopharmaceutics book, right? So that book has hydroxychloroquine and chloroquine as an example of very high volume of distribution of drugs. So the point I'm trying to drive home, that this drug remains in the body for a prolonged time, doesn't get out of the body. And if the metabolism is not happening in the right way, you can imagine the levels are going to go very high. And different agencies, for example, US FDA has put some caution, telling that there is a risk when you use this drug outside the clinical trial or hospital. American College of Cardiology has put a warning about hydroxychloroquine, and of course FDA put multiple warnings. However, ICMR, our own Indian Council of Medical Research, have found some benefits in hydroxychloroquine use. Of course, it has to be validated by actual data, which I know we are still early. We are trying to see data from agencies, but there hasn't been any explanation, clinical trial explanation yet, why we are seeing death from cardiotoxicity. What is the explanation of cardiotoxicity we are seeing across the globe? So be it FDA, be it ICMR, there hasn't been any explanation. And if you look at limited data which came out so far on hydroxychloroquine PK in COVID-19 ICU patients, look at the PK. It is all over the place. And if you look at the x-axis, you are going to see all the hydroxychloroquine concentration. And look at the variability. Look at the variability of patient PK profile. And remember, these people are already very down in ICU, right? They are struggling with their life, on ventilators probably. So, and on top of it, hydroxychloroquine PK is about 4-fold difference, which is a very narrow therapeutic index drug. It is considered a narrow therapeutic index drug, means a slight difference in the concentration can eventually lead to a catastrophic effect. So this is real data from COVID-19 patients. Now the question is, as a pharmacist, pharmacologist, or pharmaceutical community, we should ask, why this variation we are seeing? Because we are also hearing, right? Like ICMR said, there are some benefits. In here also, we found some benefit. Why there is a difference between cure versus death? What is there any difference in the metabolic capacity of the individuals? What is their CYP2D6 status? Are there any pre-existing conditions who are dying or from the toxicity of hydroxychloroquine? Are they on some co-medications? These are all the questions need to be answered before we realize the effect of hydroxychloroquine. Because right now, it is a gamble we all are playing. It may work, it may not work. But the questions I'm putting here, it might allow us to answer some of those. Now, if you look at this diagram of this table of metabolism of different beta blockers, again, a very good class of drug, very, very effective drug, is metabolized by CYP2D6, several of them. So by now, we all know that hypertension is a risk factor for COVID-19, right? Hypertension is a major risk factor. And my next slide highlights that. This is from the patients of New York, which was an epicenter until last month. And now Florida is epicenter, by the way, where I live. So this is from New York patients. It shows hypertension was the major factor. More than 50% people had experienced COVID-19 with hypertension. That means those people will also be on one of the beta blockers, likely be on COVID-19 or beta blockers. Now, the problem on the other side also exists. I said earlier, hydroxychloroquine is metabolized by CYP2D6. At the same time, hydroxychloroquine also a CYP2D6 inhibitor. So I hope you are connecting the dots. So hydroxychloroquine can also affect people who are on comorbidity, means people who are on beta blockers, and they can block the metabolism of 2D6 and block the metabolism of atenolol or carbidopa, all these beta blockers that can lead to increased concentration of beta blockers, means hypotensive effect. The patient is already in a very low energy state in ICU or in the hospital. And if the patient is now hypotensive, the vital parameters are going to stop, right? So hydroxychloroquine is not just toxic to the heart by its pharmacological action. It is also a blocker of 2D6. And if you look at the effect on metoprolol, here in this table, the second column tells you area under the curve of metoprolol with placebo, means no hydroxychloroquine. But look at the boxes where I have put a rectangle here. You can see the increase in those patients when they were given hydroxychloroquine, and their numbers went up. The AUCs went up because their metabolism is not happening. The concentrations are going up. As a result, they are going to experience hypotension, means lower blood pressure, and that can lead to finally blockade of cardiac arrest or finally blockade of vital parameters of the patient, and that means death. So I think there are research opportunities, I feel, and I am hoping that this conversation is going to generate some interest, because right now COVID patient and treatment is now happening everywhere, including the part of Maharashtra and India. You are right now happening everywhere. It opens up research opportunity for us. First of all, we need to know the pharmacogenomic profiling of CYP2D6 in COVID-19 patients. What is their profile before we give them hydroxychloroquine? In case we give them, we need to do therapeutic drug monitoring, or TDM. What is the concentration of hydroxychloroquine in those patients? And then finally do a PK-PD relationship, pharmacokinetics, pharmacodynamic, means how are their vital parameters responding to the concentration of the hydroxychloroquine, and cardiotoxicity. How are their cardiac parameters? There are several markers now, troponin or ECG, several enzymatic markers of cardiotoxicity. And then we need to establish a relationship between obesity, because it will affect the volume of distribution, hydroxychloroquine effect, and cardiotoxicity. Is there any accumulation of the drug in the heart? That's why we are seeing more cardiotoxicity. And then finally, what is the effect of this drug on comorbidity drugs? Means I mentioned cardiovascular was one, but it can also impact people on antidepressants, people on analgesics such as morphine or codeine for pain purposes or cough purposes, right? So all these questions need to be answered for 2D6 or hydroxychloroquine before we can comfortably use it. And in terms of Indian context, I think even it's more relevant, because India has a very diverse origin. If you think about how Aryans and Dravidians and other origins or Mongolian origins India have, the northeastern part of India, western part of India, or southern part of India, or northern part of India, they could be very different because of their origin of like 100, 200, 500 years back. And that opens up genetic stratification opportunity for drug metabolizing enzyme and to identify drug disease interaction. So how different diseases are affecting the pharmacokinetic outcome. And finally, the PGX, or Indian herbal compound metabolism. India is one of the resource center of the world in terms of natural products. But a lot of times we hear about negative outcomes, unfortunately. Is pharmacogenomics contributing to these outcomes? If we understand our Indian population in a better way, I think the use of herbal compounds will be more effective, more beneficial. But again, it goes back to the idea of understanding their ability to metabolize any drug or compounds in the body and how it affects the outcome. And as a country, I must tell that in the Indian context, there is a huge opportunity. And as an academia or industry, in both cases, we need to debate what is the best way to put our outcome to the healthcare resources. And because of India's huge market and diversity for drug therapeutics, the repository of human genetic diversity is important. We need to start developing a data bank or bank of genetic compositions, so that the research in the academia or in the industry could be driven by that. The clinical trials can be driven by that. And that is going to eventually help to minimize the cost of healthcare, as well as expedite drug and health or herbal outcomes, herbal product outcomes even better. With that, I'll finish off showing a couple of slides, which is around my surroundings where I live. Florida is very much like India and pretty much like actually coastal Maharashtra and Mumbai. You can see a lot of coconut plants and so on. These pictures are either behind my back house, the backyard. Also, we have some nice iguana kind of very diverse animals. And fauna and flora of this part of Florida is really fascinating and very much like in some parts of India. So I really enjoy the nature here, the natural component of this part of the US. And finally, thank you for your patient hearing. I might have gone slightly over time. So I'll be happy to answer any question or for any discussion you may have with the topic or anything related to pharmaceutical research. Thank you.
Thank you, sir. It was a very informative session.
Thank you.
Sir, we're just getting some questions.
Sure.
Yes, Chandan Tiwari. Chandan Tiwari.
Sir, my question is it possible to cure hereditary disease by its roots, and how?
Hereditary diseases are, to be honest, is difficult to treat by this approach because for example, hereditary diseases, you cannot change anything. What you can do really is you can manage the disease better but if someone knows ahead of time, they can take appropriate lifestyle measure or healthcare measures so that the impact can be minimized. But if it comes to drug related genetic changes from generation to generation, for example, if I know my mother has CYP2D6 polymorphism, I can be more aware of it and start testing for pharmacogenomic results, and or I'll be more careful with those drugs. But it can't be changed, right? Genetic composition is always a lifetime, and as we heredit those composition and we carry them throughout the life. So in short, those conditions can be managed, but they cannot be totally eliminated, those symptoms.
Thank you, sir.
Well,
Sir, I have one question.
sure.
As you might know that in USA, death rates due to corona are high as compared to India. So what is the role of genomics in this?
Very good question, actually. Very good question. I think there are two parts of it. One is definitely genomics, because also one other thing to notice, as far as my knowledge goes, although there is not enough data has been released by the government, as far as I understand, in US, the people from Asian origin, so far, they got relatively lesser COVID cases. So that also indicates to some extent there is a genomic role. Along with genomic, there is another component is also probably impacting is diet. The immunity level is always controlled by two aspects. One is environment, and other is genetics. The type of diet the Indian population have, the spices and lifestyle and everything, I think that is impacting heavily or helping heavily in reducing the death rate in India compared to US. So if you ask me that question, yes, the immunity levels are impacted by cytokine and enzyme, a protein called cytokines, a group of proteins, and how we react to that protein. That is, there are polymorphism of cytokines also. So that means if I have more cytokine polymorphism, or if I have a variant which cannot fight with the disease, there is likelihood I will be more prone to the death compared to other individuals. But again, there is no research. This is all hypothesis. This is, we are talking based on what we know in other similar diseases. But regarding COVID-19, we don't have any experimental evidence to say whether genomics is a reason of differences between population death and so on, different countries.
Thank you, sir.
Thank you.
Sir, I think so. There are no more questions to be from the audiences, I think. Now moving ahead, I would like to ask the
Devani
chairperson for today's.
sir,
Yes,
The
madam.
chat box.
Devani sir, there
Yes.
are questions in the chat box.
Okay, just a minute. Rutuja Cabra. Yes, Rutuja Cabra. Hello.
You read the question.
Yes. Sir, there is a question from one of our WHO discontinued hydroxychloroquine and lopinavir. So this is a question from one of our audience. WHO continued hydroxychloroquine and lopinavir. Why?
Yes. So WHO actually has, as we saw during this whole time, there was a change in their recommendation, and rightly so, because as new evidence come, or even if it is limited, we need to change the approach. Initially, there was a lot of interest about hydroxychloroquine, but subsequently, because of those deaths globally, there were deaths from hydroxychloroquine use. And I think they kept open for countries to decide whether to use. But if you personally ask me, I think the science behind that drug is very weak, number one. The mechanism for which it can target the virus. Second, because of toxicity, this drug would have been fine to use if there was no toxicity. But because CYP2D6 and it is an inhibitor of comorbid drugs, a lot of issues are there with the pharmacokinetic aspect also. So that's why, in spite of WHO kept it open for use by countries, but I think you are going to see, you are seeing already a drop in hydroxychloroquine use. At least we are seeing in US. I think in India, people are still using, if I understand correctly. And part of the reason, globally, there is a now shortage of remdesivir. I can talk about my own state here in Florida. We are out of remdesivir. And remdesivir is the drug which was shown recently by Gilead Pharmaceuticals that it can reduce 62% of the symptoms, means it can decrease the mortality by 62%. That's a huge success, I can tell. But compared to remdesivir, hydroxychloroquine, there is no such data, right? So and I'm hoping, considering India is a powerhouse for manufacturing and always pioneer in manufacturing industry, I hope India can produce remdesivir quickly for their population and worldwide, because hydroxychloroquine is not going to take very far. It's not going to take very far. I think remdesivir has much superior outcome, which is a typical antiviral drug, by the way. And remdesivir was developed against Ebola, actually. When Ebola came about 6 years back, this was developed against that drug in a very traditional drug development way, drug designing, computational drug designing model. And so they have found some outcomes. So I have more confidence on remdesivir because of its profile than hydroxychloroquine, even if WHO says the WHO keeps it open for use by the countries. And lopinavir is really never got any momentum as a treatment. So that's solely my opinion.
Sir, there is one question from Samir Lakde. Dr. Samir Lakde.
Sure.
Yes, sir. Sir, good evening, sir.
Good evening.
Your nice presentation and
you.
information, sir. So my question is, if I want to do in vitro cytochrome P450 enzyme inhibition study for the extract or plant extracts, which method is best suitable method for this study?
Sure. So I'll give you a brief answer here, but please email me after the presentation. Sure. I'll be giving you can give you more detail. I'll give for the audience and everyone's time, I'll give a brief answer. So human liver microsomes are the best model for in vitro lab studies for human extract, and you need an HPLC or LCMS to analyze the metabolites, and it's typically straightforward method. It's not very complicated. So I can give you more info because when I was in India about 8 years back, I returned India actually I think at the beginning, Dr. Divani mentioned, I returned India for good. At that time, I did some of those studies and I had identified some commercial sources where you can get liver samples, liver microsomes or intestinal microsomes. So I'll be more than happy to provide more info. Please email me in my email address.
Sure, sir. Thank you, sir. Thank you for the answer. So the second question is,
Yes.
Hello.
Yeah, go ahead, please.
Hello. Sir, how reliable are cytochrome 450 metabolism related drug interaction in non-clinical research?
So you mean preclinical research like animal model
Yes,
or
yes. Yes, yes,
yeah,
yes.
again, very good question. Usually mice and rat are very close, if not, of course, if you go to primates, higher animals like monkey or models such as dogs, they are very similar. Their P450 profile is very similar to humans, but rats and mice have been utilized heavily. I have worked with the rats, mice, primarily these two models, and their results are always uh, mimicked when it goes to in vitro human sample. The advantage of this research area is instead of going to the humans directly, which is always difficult, right? We can use
Yes, sir.
human sample, like human liver sample. We can use intestine sample from commercial sources and utilize it in the lab, and their data, I can tell you, very close to like in vivo human data, clinical situation data. So we have preclinical, we have clinical, and then we have in between semi clinical, where you are using human samples, real samples, and you use those to do in vitro studies or in situ study, we call it.
Hello.
Hello.
Sir, there is a question from YouTube
Sure.
from Saili Chaudhary ma'am. Sir, how can we control COVID-19 either through prevention or through therapy? As we see, prevention is not working. Please explain. This is the question
Yes.
from Saili Chaudhary ma'am from YouTube.
Sure. Of course, ma'am, we are all trying to uh, find that answer. I think every head of the country, head of the health agency, we are all trying to answer that. But I'm sure we all have our own approach also. I can tell my thought process. Uh, I think someone asked about the immunity difference between different population. I think the key way and one of the reasons India is doing well, like relatively, in spite of a huge population, is immunity. So I think we need to boost our innate or our inbuilt immunity by diet, appropriate diet, fruits, supplements. Like I'll tell you, uh, uh, I take vitamin C and vitamin D supplements every day for last several years, and now in last 4 months I am taking more regularly, morning, afternoon, because obviously situation is different, because vitamin C is an immunity booster, and vitamin D is also anti-inflammatory, and it can fight infection. There are enough data, we know that. And vitamin D, I have my own bias, you can tell me, because I have my research area in vitamin D, and I hope in future sometime I would like to, I'll be interested to share my vitamin D metabolism research in oncology. But the point is having our immunity strong by diet or health supplements. Number one, there is no replacement of that, because once people are infected with the COVID, I think we are looking the chances of fighting with it is very less. So we have to start even before it happens. And of course, use of mask, we all know that we can prevent it. In terms of physiologically, having a good healthy lifestyle, sleep is another factor, because sleep build immunity. I think this is extremely important, to have 7 to 8 6 to 7 hours or 8 hours of sleep to keep our immunity intact, along with diet and health supplement and exercise. This is my thought, because drugs are not going to save us at this time, and vaccines are at least 6 months away.
Yes, Miss Ankita Naval. Hello.
Yes, please.
Miss Ankita Naval. Dr. Channamar.
Thank you, sir. Hello, sir.
Yes, please.
So first of all, I must thank for today's nice session, sir. Sir, I want to ask one question. Is it true that coronavirus is changing its structure continuously, or or different strains are available there? What is true, sir?
No, absolutely. There are several, and there is no number right now. That's why I'm saying several strains, and within the country it's changing. But there is definitely a change that's between India or Europe and US, and there is an assumption or hypothesis that the strain which is working in India is perhaps slightly milder than
Yes,
the
sir.
strain in Italy or Spain and US. Although, again, it could be the immunity also, right? What I just said earlier,
Yes.
it's possible that the strain is still very virulent or very lethal, but because of the immunity of Indian population, diet and everything, it is fighting well. So, but scientifically, there has been already screened. There are several strains of these. Some of them are less lethal or virulent than the other strains. So it is true.
So, one more question, sir. So then what are the obstacles in this vaccine development, sir? How the companies they are developing their vaccine?
Right. So
against which particular strain?
Right. So very good question. So 3 companies are right now in the forefront. One is Pfizer. Other one is Moderna. You might have heard about this, right? It's a new biotech company in Boston, but it is making all kinds of news. Moderna, Pfizer, and third one is Oxford. Oxford and AstraZeneca, they have some collaboration, but Oxford University is doing it. So they are the front runners. And I just yesterday evening, I got that news. Moderna is starting phase 3 clinical trial on July 27. So now it will take probably 4 to, and they were able to generate antibodies in the 45 patients they already did the trial. Now the question is, which strain you are using? Are those strain, because in antigens and antibodies there is something called common epitope, right? Common epitope means a common amino acid in the virus which can be recognized by the antibody and target it, right? That's the purpose of vaccine. Now, if the strains, we don't know which component are making changes in the virus. Is the part which they are targeting, or is the part which it is a non critical? And that another level of uncertainty we are facing, that even if we have the vaccine, it may work for you, it may not work for me, because I may get a different virus than the population is getting. And I think this is more critical when it comes to the flu season. In US, flu season every year is an annual event, if I can say, sadly. Compared to India, that we always get a flu season between October to March every year. And every year there is a new strain coming. So that's why every year there's a new vaccine formulation happen with the industry. And even after all this, there is a large number, millions of people still get, after vaccination, the flu in the schools, in the workplaces, and so on. So my point is, even if the vaccine is developed, I think there is absolutely no certainty that it is going to work, because of viral diseases change very quickly. So our best bet is to keep ourselves fit, immune boosted, and healthy. That is the only way to fight against it. We can't rely on vaccine unless there is a medication which can treat directly, just like other viral diseases we have treated in the past.
Thank you, sir. Thank you.
Sir, there is one more question from audience. Ebola, is there any correlation between Ebola and coronavirus disease?
No. Other than both are dangerous, I think there is no similarity. They come from very different class family, I meant to say, and Ebola was more lethal actually. By the way, there were more deaths, but we never gave that importance. Unfortunately, we as a world, as a humanity, we never gave importance. Partly because it never happened in big countries, right? It didn't happen in most populous countries. Most of the Ebola cases were in Africa. And if you go through some literature now, you will see death rates were in double digits, way higher than what is COVID. But it also didn't spread to 80% of the world. So that means it didn't really hurt many people. Hopefully, COVID will also have a similar fate like Ebola. That means it will go away after 6 months or 1 year by its natural cycle, because virus has sometimes natural cycle. Except AIDS and hepatitis and some other very old ones, you see not all viruses remain on this earth. They cannot survive, right? At some point, they go through a natural cycle of elimination from the world. So hopefully coronavirus too will also have a similar fate very soon.
Sir, there is a question from Mr. Paresh Wadhwani. Is corona treated patient permanently immunized or it's for time being?
Yes, very good question. I think so far what we understand, very limited data coming out. We cannot. It is uncertain. It could be temporary. The data so far is very ambiguous, and there is no indication that once the person getting infected, they're not going to get it again. And as a matter of fact, some cases already appeared in Europe and in US, especially Europe. Most of the times, they got reinfected. Which is against the immunology concepts, right? Vaccine concepts. So at this time is unknown. And if it is the case, that will put a huge question mark on the success of the vaccines, right? Because that means having antibody doesn't mean much, and maybe that goes back to variants or mutants. I got vaccinated for probably 10 variants, whatever is known today. But eventually there are another 10 variants or mutant came to the population, and I get one of those. I may still get it, depending on the dissimilarity or difference in their amino acid sequence, right? So in short, it's unknown right now, but likely people don't develop. People don't develop the disease again. But we have to wait. It's very early, probably.
Sir, one of the Dr. Shilpa Gawande has given a question. What is your opinion on plasma therapy?
I don't, to be honest with you, I don't have much opinion about that because I largely focus on the drug area, and within that pharmacogenomics and drug outcomes, drug treatment outcomes. I think this is a similar concept where you generate antibodies and you try to treat. Instead of vaccine, you are giving an individual blood or plasma with antibodies, right? Similar concept like vaccine. There has been limited success, I have heard, but I'll acknowledge I have limited insight into that component. But it could work just like vaccines are designed for, right? Instead of giving the vaccine, we are giving already made antibodies to the individual. So it is a potential therapy, but I don't have much opinion about that.
Thank you, sir.
Sure.
Hello.
Yes.
Hello.
Yes, please go ahead.
Good evening, sir.
Good evening.
Sir, thank you so much for your excellent session. It was very nice and informative.
Thank you.
So
Thank
my
you.
question is how to control this viral diseases like Ebola or coronavirus? How to control, how to prevent this type of diseases?
So I think it goes back to the previous similar question that one thing is, there are two parts of it. One is a global picture. I think you can see when we were young kids, bacterial diseases were the major issues, right? We used to get bacterial infections all the time, and even our parents said that even in their time. In their time, there was issues with a lot of other. Just give me a second, please. Let me, yeah. Yeah. In earlier days, the bacterial diseases were more challenging, but in recent times, you see. Let me if I can just.
Hello.
Okay, sorry for that. So I think we have in last 40 years we have shifted to a different dimension altogether, and part of it potentially contributed to global warming. And because viruses, I think you see that for last 20 years we got 5 major outbreak, either from SARS coronavirus family or from Ebola like of diseases. So part of it is how we are destabilizing the environment, how we are destabilizing the ecology of the world. So instead of the bacteria, now viruses are more prevalent, and so that one way is more a global effort, means how we can, which is difficult, and unfortunately it is going backward instead of progression that global warming need to be addressed, and more green and other ecological factors need to be considered. The second part of it is our lifestyle. It has changed. If you think about it, our diet, our lifestyle, our sleeping pattern, our digital life, our electronics, we are surrounded by electronics all the time. People have a very different sleep pattern, diet pattern. We are slowly kind of forgetting indigenous foods, especially in bigger cities, that is a more of a larger problem. So all these are contributing to eventually our ability to fight. We have to all understand microbes were always there, and microbes are there around us. It is our ability to fight it. And because microbes undergo some genetic changes over time, so they either become more virulent or lethal, or they go away if they can't get into our body and survive, right? So I hope as a human being in future, we take care of the nature, number one, and we take care of ourselves. I think that's the way we can fight it. We can build immunity. We give less opportunity to the virus to develop in the environment by having more ecologically friendly environment, rather than focusing on more development. Unfortunately.
Thank you, sir.
Yes, sir. Thank you so much.
RF Kamil.
Hello.
Hello.
Sir, can you hear me?
Yes, I can hear you. Please.
Thank you very much for delivering such a nice session, sir. I do have a question, like how much time will it take for personal medicines, like studying individuals' genetics and designing particular and specific medicines for that person only? So with this current rate of development of technology, what do you think? How much time will it take for this personal kind of medicine to become very popular and common like there are blood tests. So we take a blood sample and then we prescribe medicines for that. So with this particular rate of development of technology, what do you think? How much time will it take, and how feasible will it be? Will it be cost effective?
Right.
Or
So
what
again,
would
very
be
good.
the future of this?
Sure. Very good question again. The genetic information or pharmacogenomics can be used used in two ways. One is by drug development teams, be it academia, be it industry. Means if I know a certain disease has mutant version, and there are drugs, by the way, and like I said, last 10 years, 15 years, we made tremendous progress in this area. There are some anti-cancer drug for colorectal cancer and lung cancer. Those are actually developed against mutant version, not the wild type version. So those were developed against muted version. So this is only for people who have a different sequence than normal people or common people. So that is one way to approach. And the second way is utilizing pharmacogenomic information for patient outcome, which you described. And I think your question is more in that line. So I think it is already in practice in US and in India in a very limited way. And I'll say very, very limited way. But in US, what we have is now these guidelines. I showed some of them in the slide, CPIC guidelines. Those tell, and even in FDA levels, if you look at the FDA package inserts, they tell actually that, okay, this drug has very strong influence of pharmacogenomics. So the patient might be or must be tested before they are administered the drug. So there are warnings in FDA package inserts already. So now the point is the next part, cost, because there is a cost involved from sampling to sequencing, and that is the part US is still be divided because this cost has to come from the insurance or from the patient. But I believe US has come a long way in budget in terms of supporting those tests, but in the long run, you are saving money in the patient adverse effects, right? Less hospitalization, less drug failure. So I think India, country like India, where a huge part is also a government cost, right? US is heavily privatized healthcare sector. India also privatized, but a major initiative is also run by government hospitals, right? So that means government money is flowing there. If we have a process and technology in place, initially it will cost money, but down the line, the government is going to save a lot of uh, 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 revenues. Think of Aadhaar card, right? When Aadhaar card started, a lot of money was needed because everywhere you have to have this machine for scanning biometrics. But now you see government is saving money because all the benefits they are giving to the individuals directly, right, in their bank account. So this is a lifetime change you are transitioning. So there will be some investment from government, and it has to start with small, but eventually the government or private sectors are going to get the benefit in terms of patient treatment outcome, or be it drug development success, because a lot of clinical trials fail of toxicity or not working the drug, and it's partly because those people are not meant for their drug, right? Because those people genetically are different, they may not work, and if we know this before the trial, we may not even start the trial on those people.
Okay.
So it is cost, it's a bit of a cost at the beginning. It is getting cheaper and cheaper with the technology developing, and India being a powerhouse for science, diagnostic kits and a lot of things. I think it will not be difficult to implement if Indian companies pick it up seriously.
Okay, thank you very
sir.
much.
Now I request Dr. Madia sir to share her views on the today's session. Dr. Madia sir, please. Hello. Madhya sir.
माइक अनम्यूट कीजिए.
Hello.
सर माइक अनम्यूट कीजिए. बढ़िया सर.
हाँ
हाँ,
हाँ हाँ कीप. Okay. Good morning, Dr. Subrata.
Good morning.
Sir, in the ongoing pandemic times, the topic which you have highlighted and discussed becomes extremely relevant and worthy of giving thought of. So the way you have simplified this complex topic for us was absolutely incredible and helped us connect to it so well. I am sure all of us present today in this virtual meet echo my words. Dr. Subrata, you kept us scaled up to the topic throughout the presentation, and we have had a good insight about the topic. You have explained very nicely how pharmacogenomic testing helps to prevent gene-drug interaction. One size does not fit all. We have had this opportunity to hear about how we can use pharmacogenomic as a tool and explore its potential to tackle with COVID-19. From the core of my heart, I would like to thank you, Dr. Subrata sir, for your kind presence and enlightening us. Thank you very much.
Thank you, thank you, Dr. Madhya, for nice and kind words. And I am really.
Dr. Subrata sir, I am KMK alumni. I passed in 1983 from Bombay. KMK.
I see. Okay, KM Kundrani. Yeah, of course, we went several times to write my university exams, right? KM Kundrani used to be our site of exam for annual exams.
Dr. J.K. Lala.
Yeah, Dr. Lala. Yes.
Yeah. Thank you very much. Thank you very much.
Thank you.
Thank you, sir. Thank you very much. Now I request Ms. Prajakta Bansal to express the vote of thanks.
Hello. Am I audible to all of you?
Yes.
Hello. Very good evening to all of you. First of all, thank you so much, Devani sir. Dr. Subrata Deb sir, we are so thankful for your valuable time you gave to us on behalf of management, principal sir, and entire team of PWCOP College of Pharmacy. I express my deep sense of gratitude for your valuable time and knowledgeable information in such a pandemic situation. Thank you so much. Moving ahead, I would like to thank Principal Sir for arranging such a beautiful webinar series in such situation. And I would also like to thank Dr. Nitin Kochar Sir, Professor, Department of Pharmacology, P. Wadhwani College of Pharmacy, and Dr. Abhijit Srirao Sir, Assistant Professor, P. Wadhwani College of Pharmacy, Yotmal, the Associate Coordinator of webinar series. Again, I would like to express my thanks to Dr. Moale Sir, who is a session coordinator, and respected Devani Sir, who is also a session coordinator, for arranging something like this in very smooth way. Thank you so much, everyone. And last but not least, I would like to thank all the participants for their kind cooperation. Thank you so much. We will meet over to Devani sir.
Thank you very much for today's session. We are concluding the session today over here. Tomorrow there
Before
will be
that, I personally have to thank Dr. Subrata Dev for your presentation in a nice way. Actually, my battery is very low, but I came in my car to charge it and listening you. I'm sitting in my car and listening you.
I can see that. Thank you.
So,
Thank you for the effort.
So it's very informative. I am taking much interest in that. So thank you, sir, for your nice presentation. Thank you, Madhya sir, to be the chairperson of this session. Thank you. Thank you very much. Thank you all. And Subrata Dev sir, be in touch in future also. We will arrange so many seminars.
Good. So if I can one last comment here. first of all, thank you everyone. Dr. Chandevar sir, Dr. Kocher, Dr. Srirao, and everyone involved here. I must tell this. I feel connected. I feel connected even, and I should also tell this. I was supposed to be in India actually today, and I was supposed to leave for US. I had my trip from June 4th to July 15th. Already booked and everything. Then I had to cancel for the COVID. And today was the date I was to fly out from Mumbai, actually. So it's a very coincident that we are connecting today and having a conversation, and it gives me immense pleasure, and I feel at home while talking with you all. Thank you.
Thank you, sir. Thank you.
Sir. Thank you so much.
Thank you.
Tomorrow we will be having a session at 9 o'clock again.
So I'll take off.
Thank you, sir. Thank you, sir.
All participants should join tomorrow by 8:45 a.m. Thank you.
Thank you.
चार्जर काही काम नाही करत, घेऊन येतो. चार्जर काही काम नाही करत. कुठला चार्जर आणून ठेवून दिला माहिती आहे. इथला ओरिजिनल चार्जर कुठे गेला तो? थोडी आहे. एकच बनवलं ना सगळं. सरांनी विचारलं इतके पैसे है क्या, पण 6 लाख कर रहे हो ना ट्रांसफर, रिटर्न जब आएंगे तो क्लियर हो जाएंगे. एकच बनवतो का तू? आणि रेकॉर्डला कुठे 2 वर्ष आहे गेले मी तसं. एक वर्ष आहे. कुठं सरांची आता इथं एकत्रित करायचं, शिफ्ट करावं लागेल. हे चालत नाही, रेकॉर्डला येत नाही ना मग. रिटर्न से सांगा दुसऱ्याला. सांगा सरांचं घरी जाय दिला वाटतं. ठीक आहे. सेल्फ हा नाही 50 बोललो नाही. आय कार्ड वगैरे आहे ना तुझ्याकडे? चेक करतो. चेक करा सगळं काढून क्लीन कर आणि सॅनिटाईज कर सगळं.